thanks a lot to the organizers for this truly amazing conference. It's one of those few times when multidisciplinary really works. I have learned so much. It's really exciting. And so today I'm going to talk to you about how functional near-infrared spectroscopy may be used to image the developing brain, so young infants. After the previous amazing talk, I don't really need to introduce this in great detail, and many of you know more about this than I do, but the technique of near-infrared spectroscopy originates in the fact that we have this optic window. So you see the extinction coefficients of all those compounds that play a significant role in biological tissues, oxygenated and deoxygenated hemoglobin, melanin, water, et cetera, and you can see that our tissues are transparent, quote unquote, to infrared, visible red, and near-infrared light, as you can see in the illustration. And so this has been used in the literature for almost about 20, 30 years now to image the human brain, in particular the infant brain, using a visible red and near-infrared light in the following way. We have a light source so this is the scalp surface. We have a light source that we shine on the head. It goes through this banana-shaped trajectory, and we measure the exiting light, the intensity of the exiting light a few centimeters away. And we get a measurement channel, so a near-infrared spectroscopy channel here. And of course, we can have multiple channels across the scalp surface. And we can measure, measuring actually the light intensities, we can calculate or compute the concentration, or rather the concentration changes of the two chromophores that interest us because they're related to neural activity, in particular the concentration changes of oxygenated and deoxygenated hemoglobin. And so in most of the applications, I'm emphasizing the fact that we are measuring concentration changes because in most of the commercially available NIRS machines, scatter and the differential path length factor are unknown, and so essentially we are only measuring relative and not absolute concentrations. And so, as we already saw in the previous talk, we get the hemodynamic response function, so an increase typically in oxygenated hemoglobin and a decrease in deoxygenated hemoglobin. So this gives us functional near-infrared spectroscopy, which among all the brain imaging techniques can be sort of found in this space where its spatial resolution is not extremely high compared to, say, MRI, but it's definitely not subject to the inverse problem, unlike EEG electroencephalography. So at least it's accurate. Its spatial resolution is accurate. Its temporal resolution is relatively low, but this is not a problem of the technology. The machines actually sample fast. It's the hemodynamic response itself that builds up over several seconds, unlike, again, EEG, which measures in the millisecond range. And the third dimension, and this is what's particularly relevant for us developmental psychologists or developmental neuroscientists, is this third dimension here, which is the degree of tolerance, or how easy or nice it is for the participant to be in a study. And so you see this is also color-coded. MEG, PET, or MRI are not quite suitable for small children or young babies because they are uncomfortable or actually quite invasive, say PET with the isotope. While NIRS and EEG are two techniques that are very comfortable even for young infants to participate in. So all in all, functional near-infrared spectroscopy, when applied to active, typical, so non-clinical human populations, has quite a few advantages over some techniques. So as we discussed, it has not very high but accurate spatial resolution, low temporal resolution, but it is completely non-invasive. So this is really a huge advantage when we are working with babies or small children. It's very inexpensive, and that's, again, a huge advantage compared to many of the other techniques. And I'll come to this, I will illustrate this with pictures, but it's wearable and portable, so it's very well suited for bedside applications. So both in clinical settings where, say, a participant is linked to life support, so you can't transport the participant, you can't take the participant to have an MRI scan, hand, but you can take NIRS to the participant. So it's very well suited for these kinds of settings. It's also really easy to learn to apply, so it doesn't have a really steep learning curve for the experimenter. And it's a silent technique. There's no gel, no carrier substance. It's really comfortable to use and to apply. And again, very, very important for child or developmental participants. It's not very sensitive to motion artifacts. And so given these characteristics, many advantages, I want to illustrate the use of FNIRS in adults and then focus on infants. So indeed, in adults, FNIRS is what we call a niche technique, right? So if you can put an adult into an MRI, which is most of the times, you will do that to get the extra spatial resolution, or if you're interested in a higher temporal resolution, you would do EEG. So most of the times, FNIRS is not used with adults, but there are exceptions. So these are the niches in which FNIRS is very well suited for imaging, in particular in behaving participants. It's very often used in sports science because it's very motion tolerant and it's small and portable. So you can have athletes running around with NIRS on their heads, or one of my favorite applications in France, there's a lab that uses it in pilots that fly planes, because the human error, of course, is one of the most important sources of aviation accidents. And so they send pilots up with NIRS on their heads, and that's really cool. And again, bedside applications, right? So in adults, you can very often use NIRS to, or people very often use NIRS to monitor stroke patients or coma patients. So computer brain interfaces are also quite common with NIRS. But our focus is on developmental populations. And that's because, as we were talking in the last or the previous presentation, essentially depth or penetration is a very important question here. If you remember the banana, how deep your banana is will determine how deep you go into the brain. And because infants and children's tissues are thinner than adults, actually NIRS penetrates a lot deeper into the head, and so can image almost all of the cortex in a child or in an infant, whereas it really just reaches the surface of the cortex in adults. And so given this particular advantage, it has been used in infants very actively in the last 2 or 3 decades. Our group focuses on applications in mostly in typically developing newborn infants. And when I say newborn, these really are babies at the hospital a day or 2 old, or infants the first couple of years. It's perfectly safe, so it's very often used with preterm infants, because obviously brain oxygenation is a really important issue for survival and outcome prospects for preterm babies. And so it's used in bedside monitoring in that population. And as I will also illustrate, it can be used with cochlear implant users because it doesn't interfere, or rather the cochlear implant doesn't interfere with the NIRS signal, whereas it does with the EEG signal, for example. Or it's very well suited for participants who don't like social interaction, so who would have a much harder time with a human experimenter, but can sit around and watch things on a computer screen like autistic children. And so just to give you sort of an illustration of what this looks like, these are newborn infants, so really day 1, 2, 3 at the hospital, and you see that they are quietly sleeping through the study with the cap, the NIRS cap on their heads. It's another brand, another type of NIRS machine, again, a newborn infant. These are somewhat older infants. Again, you see that these are stretchy caps, a bit like swimming pool caps, and they're just sort of happily sitting through the study. And remember that NIRS and EEG have opposite strengths and weaknesses. So EEG gives you high temporal resolution, but not good spatial resolution, whereas NIRS is the opposite. If you combine the two, you get best of both worlds. And so here what you can see is a combined NIRS EEG recording. These are the NIRS optodes here, and these are EEG electrodes with the little shiny lights on them. So this is what it looks like. And then one thing, as I was saying, that's important is that it's wearable and portable, and that's a huge advantage when you have to work with special populations. Here we have a young deaf participant who has a cochlear implant, and these children are much more quiet and happier when they're tested at home. And this is what you can see here. The nearest machine is this here. So you see the size, it's smaller than a computer. It's like a book. You can put it in a backpack, put your backpack on your back, and just go to the participant's home. And so it's really comfortable to allow them to be tested there. They're much more quiet. We get much better signal, no movement artifacts. And here you can see Gaia, who just got the Best Poster Award, who is testing the participant. And so I would just like to sort of give you an illustration of what we get from NIRS in young babies, showing you one of our recent studies. In this study, we actually have an adult newborn infant comparison. So it sort of gives you an idea of the type of use we can put NIRS to when imaging the developmental brain. So in this study, we were interested in how newborn infants perceive cries. So of course, we all know that babies, when they're born, the first thing they do is start crying. And actually, that's the only signal they can produce, right? They can't talk, they can't laugh, they can't whistle. What they can do is to cry. Actually, very interestingly, this cry seems to already have the melody, the intonational contour of the language spoken by the mother during pregnancy. So this is a first indication that it might be related to language. And indeed, caregivers, so parents or doctors or nurses, interpret cries as communicative. They can tell apart whether it's a pain cry or a hunger cry. We definitely think that when babies are crying, they're trying to sort of call us or ask for help or something like that. So it's interpreted again as communicative. So there is a suggestion in the literature that indeed cries are somehow precursors to language in babies. And so this is what inspired our question to try and see whether babies themselves perceive cries. So when they listen to other babies' cries, as if that was really something language-like or communicative or something related to that, remembering that that's the only thing that they themselves can produce. So what we did was to test newborn infants at the hospital at birth, so age 1 to 3 days, and adults in the lab. So you see an infant participant, you see an adult participant, and roughly the localization. So we are targeting the classical auditory areas in the temporal cortex, frontal and parietal areas. In particular, some of these areas are also involved in production. And so what we tested them on was speech in French. The babies were born in Paris, in France, so their native language, the language they heard prenatally, was French, and cries of other, not their own, but other French newborn infants. So I'll just give you sort of a...
Tu vas avoir un cousin ou une cousine.
This is a French sentence. Okay, the others don't work. And this is cry. Okay, so they listened to speech and cries. Importantly, the adults were tested here in Padua, say they were Italian, so they did not understand French. And the point was to make them similar to newborns, right? So they hear something, acoustically they can hear what it is, but they don't understand it, right? So they can't process it really as their native language. They're not showing any processing of the meaning or the grammar, only of the sounds. And so our question was whether babies and adults process speech and cries differently, because of course adults produce speech most of the times, and newborns can only produce cries. And so what we did was to indeed compare their hemodynamic responses, and here is what we got. First, the adults, and the first thing to notice is how flat the responses are. This is precisely because the adult head with its thicker tissues just absorbs a lot more light, and our signal is weaker. But what you do see here is that both in the left and right hemispheres, so the localizations of these channels are colored here, right? So you can find them, you know, on the brain or on the head surface, kind of. We find a greater response to speech as compared to cries. So adults respond more to speech, and that's indeed the signal they're used to, that they're familiar with, and they can produce. So far, this is not particularly surprising. The nicer result was the infant result. So newborn infants actually respond more to cries, right? So you see that here in the right temporal areas, we find a much greater response. You see the oxy response being much higher to cries than to speech. And so this very nicely, and you see the amplitude again, just to go back to the previous point, the amplitude of the responses is much greater because indeed their thinner tissues allow us to get a better signal. And so essentially the study showed us that you can meaningfully use NIRS to ask neuroscientifically or cognitively relevant questions about the infant brain. You can, despite the fact that NIRS is a niche technique with adults, you can use it to compare infants and adults. And indeed, we found this functionally very relevant difference between the two populations, both responding more to the type of communication they can produce. So I hope I have convinced you that you can use NIRS to image the newborn brain or the developing brain in general. And maybe if I didn't do a good job, you attended the posters during lunch and the coffee break, because four of my wonderful colleagues have presented, and Gaia did get the Best Poster Award. So thank you very much for your attention.